right. Thank you everyone for coming. We're going to start on time since we have some people that is following us. Hello. I'm a super happy actually for hosting Dr. Emerson Ponte today here at Purdue. For those that doesn't know, he was actually my former advisor in my master degree, and he also followed on my PhD when I was doing Brazil. So you're going to see part of what he did in the past of those years. So his topic today, actually, he's going to present about the 20 years that he works in Brazil related to Fusarium and everything else that is together. So he's going to go over those. But I also want to mention that, so he's originally from Brazil. He works from first time we met when I was on the undergraduate, so was on the University of Rio Grande do Sul. So it's the last state in the very south state in Brazil. And then in the middle of my master degree, he decided to move to University of Viçosa, where he's currently working. He's a full professor, and he's been there for 12 years. But he also worked in the U.S. in the past. So during his PhD, he came to Cornell University, where he did a part of his PhD, and then he came back to the United States in Iowa State for postdoc position. So he knows very of us in the plant pathology, and you're going to see all the collaborations, but I think that's what I can talk about now. So I think you can start talking about your presentation, but thank you for coming. And he actually is doing a big trip to U.S., so that's very nice. And I'm happy that you started. So we're going to be happy to hear you.
Thank you very much. Good afternoon. This talk is about, it's going to be a challenge because it's how to summarize 20 years of research in 40 minutes. It's going to be a challenge. I hope you stay awake after all that time. Hope it's fun. It's been a fun journey for me. I started as of actually 20 years ago, and actually I started working with Fusarium during my PhD, doing the modeling work. And then I fell in love with this pathogen. It's a fascinating pathogen system, and I built a program, my research program, using Fusarium as the chief project over these 20 years. Then I moved to Viçosa, the center of Brazil, 12 years ago, and we had another challenge, which was another wheat disease called wheat blast. So Fusarium head blight is important in the south, but wheat blast, which are quite similar in terms of symptoms. You're going to see some photos here. And I started to work more on wheat blast. But there's a lot that has been done in Fusarium, graminearum, and other species complex during this year. Just would like to mention some of my U.S. colleagues who have collaborated with me along these years. Todd Ward from USDA and Martha Vaughan from USDA. Gary Bergstrom was the one who received me in Cornell during my PhD, and then he was able to receive 2 PhDs from my lab. He hosted them after 10 years. And then Dennis Shaw from Kansas State, also Pierre, these are the students who benefited from these interactions. Lisa Valencourt from University of Kentucky, Dave Schmale from Virginia Tech, John Leslie. We organized the Tropical Fusarium Workshop, which is the same as the Fusarium Workshop, and Paul Asker from Penn State University. Some of those people, they were analyzing data for us, they were receiving my students, so it was an opportunity to interact more and get a training for these people. So it's very strong collaboration between US and Brazil, my lab specifically. So head blight is a major disease for wheat, as you know. Still in the US, it's very important in many countries around the world. The top 10 fungal pathogens is listed here. And in Brazil is very important also, although we do not export wheat, we need to import a lot of wheat. We grow wheat in these regions, in the south and in the center of Brazil, the central western part. Here is more, is both rain-fed wheat, but this is a subtropical environment and tropical wheat. So we have challenges in these two regions. We have some high risk for Fusarium head blight in the south, and warm and cooler, for more favorable for head blight, and here in the central western, more favorable for wheat blast. Because here they plant wheat after the summer. It's after the summer crop, so it's after the summer that they grow. So it's really humid after the summer in this time of year. In some areas we can find both diseases, like you can see in this photograph, the same field and the same head. You can find both diseases, head blight and wheat blast. In the transition between the subtropics and the tropical area, we can find both. In some years, when we have a favorable storm for these diseases, we can find them. But the most important in the south is head blight, which is not important only for wheat, but also for corn and also for rice. I think your master's was using rice. No, your PhD, right? Yeah. We were exploring the species complex from rice in Brazil for the first time. So I want to summarize in 6 chapters actually, this program. The chapter number 1 is going to be a new old problem. We use a lot of different tools to answer questions about, is this a new disease? Is this re-emergent disease? What are factors contributed to the emergence of head blight as a major pathogen in the region? So we started to collect isolates to know better our enemy in the 2nd chapter. In the 3rd chapter, the food is in danger, so using chemistry to analyze for mycotoxins. And the 4th chapter was exploring the pathogen biology, the species complex, what kind of toxigenic profiles those species would present. And in the 5 chapters, naturally, we want to explore ways to combat this enemy, fungicide efficacy and economic analysis using the weapons, and is there any failure ahead? So fungicide sensitivity, that's another question that we can ask. As long as we have the isolates, we can do many different things. So that's the kind of the movie that I'm gonna show you that has 6 chapters. Chapter number 1. So we have this enemy that started to become more frequent after the 90s. So what happened after the 90s? Was it because of no-till, or was it because of climate change, or climate variability? So we started to see this kind of yield losses increasing in this after the 2000s and the 90s. We used a modeling approach to answer that question. We built, during my PhD, I built this model, simulation model for predicting the risk of head blight, and we validate this model for experiments using experimental data with high disease levels, and you can see that the simulation was pretty much close to the observations, so the simulator data was close to observations for high disease and also for low disease. It's basically a weather-based model. So every time you have the flowers, if it's warm and wet, you have a favorable environment for infection. And then you can see that there is no much risk here because it's drier. It's not so wet, not so much rain, the temperature is not so high, there is no infection. Okay, it's a dynamic simulation model. And we used this model to reconstruct the time series of risk in Brazil. And the model does not use inoculum. The model just uses weather. Okay, so the question is this because of no-till or is this because of weather? So just using weather, we can find this pattern that the disease was important during the 60s, and we could find reports in the literature in Brazil. Yes, the disease was important. So many people from Japan was coming to Brazil by the time they studied Fusarium headlight. And then during the 70s and the 80s, people at Embrapa trigo, Embrapa wheat, the disease was not important. And the model reconstructed that time series was showing that yes, the risk was lower. But after the 90s, what happened? People were blaming on no-till. And we could show that actually using weather, the weather changed. We call these a decadal change in weather. So more humid and dry, and then humid and wetter and warmer in these years here. So the research of this light, we could hypothesize that that's because the weather is changing. And that's also the case for many other places like here in the U.S., I think, because of the weather is changing. There is also change in the cultivars, change in the management, many other factors. But a weather-based model was suggesting that weather was a key player in the research of this disease in Passo Fundo, the location we used. So the other question we had, we needed to estimate the impact. So we took fungicide trial data, and we could use meta-analysis to predict the impact on yield based on FHB index. So the more disease we have, the lower the yield. So we could build a model to predict this. And then we could get these equations for a situation of high yield and low yield, and to predict the impact. So the percentage, the damage coefficient for head blight under high yield context, it's 1% yield loss per 1% disease severity. So it's pretty high relative damage coefficient compared to foliar diseases of wheat, for example. It's much higher. And we also have this damage coefficient for low yield. We could reconstruct this using a crop simulation model, using different planting dates in the same year during 28 years, and we could estimate the yield loss. And the numbers are quite similar to the ones reported in experiments. So we use a model to predict the disease, and then another model to predict yield loss. And then we have this increase during these decades, showing that yes, the yield loss is much higher due to Fusarium head blight. Okay, that's chapter 1. And chapter 2, we need to know better this enemy. And we know this, the rise of a species complex. Fusarium graminearum is not a single species, it's a part of the complex. In the past it was called lineage 7. Sensu stricto, but there are so many other players here. And we wanted to know, nobody has investigated in Brazil what was going on in terms of the species of the complex. We were just calling Fusarium graminearum. Everyone, every part. So we started to use some new tools, molecular methods. A collaboration was really important with Todd Ward from USDA. We were sending DNA to his way, and he was analyzing several isolates. And that's important. So why is it important to determine the species? Because the species, they are connected with the profile, the mycotoxin profile. So Fusarium graminearum is a potent DON, deoxynivalenol producer. And other species produce different mycotoxins. So if we have something different than graminearum, we should know the toxigenic potential of that species also. Not only the species because of the management, but also for the risk for our food. Okay, that was our goal here. And yes, we could find that graminearum, Sensu stricto, was the main player, but there was a fraction, not so small, here of Fusarium meridionale, which is from the south. Meridionale means south. Okay, and we could find meridionale in wheat, in some regions, more in Paraná State. And it's interesting because in Paraná State there's more rotation corn and wheat. Okay, less here and more here. Okay, and this is interesting because all Graminearum from Brazil, they are 15A DON. And all Meridionale from Brazil, they are nivalenol. So it's a different mycotoxin. So we should be concerned not only about DON, but also NIV, the mycotoxin. Okay? Then another study, we extended to Paraná State, higher number of isolates. We could find the Graminearum species complex here, but also different complexes from different complexes, other Fusarium species in wheat and barley. So it varies depending on the year. We have different complexes, but it's still Graminearum species complex is the main one, as you can see here in the green. The main pathogen causing being isolated from grains. And within the Graminearum species complex, still Graminearum is the main one, as you can see, but we could find Meridionale as the second player, but also Cortaderia in some other places, some other years, different years, and Austro-Americanum. So we have, I think Brazil and also South Africa and China, there are different species of the complex. I think here in the U.S. is more uniform. We have more Fusarium Graminearum as the main species, but we have more. And that's interesting because they have different chemotypes, the ability to produce different mycotoxins. So the next natural question would be, is our food safe? Chapter 3, we needed to collect the grains from commercial fields and analyze for DON and nivalenol because we have Meridionale, which is a very important pathogen species also. And we could find DON and NIV. So NIV was present. And nobody was looking at that. Nobody knew. We were the first one to report nivalenol in the south, Rio Grande do Sul state. So it was pretty random the presence of NIV in this area. So who's the bad guy? Okay, we have Graminearum, and we have in lower percentage Meridionale. What explains this dominance of Graminearum compared to Meridionale? That's the next question. I think Camila was involved in this work during her master's. And we could find that Graminearum, these two guys here, two different isolates and two different isolates of Meridionale, Graminearum is more aggressive. So if you inoculate, that explains part of the dominance of Graminearum because it's more aggressive when you inoculate it forward over time, the spread on the ears, on the spikes. And here the same. So we can see that Graminearum is more aggressive. If we look at the symptoms, they are different. So here you can see that the whole spikelet is infected, it's symptomatic. But here for Meridionale, there's a distinct pattern. Not the entire spikelet is affected. So the severity is lower. So there is something here that prevents the pathogen, or the pathogen doesn't have the mechanisms to infect more than Graminearum. And also this another evidence from my student Franklin Machado, who worked with Lisa Valancourt, also showing that yes, Graminearum is more aggressive when we compare wheat and maize. So also in wheat and maize, Meridionale here in wheat and maize, and Graminearum is more aggressive. And is there a cultivar versus species effect? Should we be concerned about potential adaptation or interaction between cultivar and isolates and species? If we select for resistance, for example, should we use only Fusarium Graminearum or a mix of Fusarium Graminearum and Meridionale? So we found that there was no interaction. So the AUDPC here for these cultivars, they are the same regardless of the isolates. They kept the same ranking. So we can use a single isolate to select for resistance to Fusarium head life in Brazil, based on our data. Graminearum is more aggressive. So we can take this one because there is no interaction. There's no cultivar that are more susceptible to Meridionale, for example. They are more susceptible always to Graminearum. Here is Camilla's work. The mycotoxin, so we inoculated different cultivars, and we wanted to quantify the presence of DON and NIF in the samples, in the spikes inoculated with both of these different isolates. That's interesting because it's only Graminearum and a mix with Meridionale. There was more DON and a little bit of NIF, but Meridionale, more NIF. So it's, yes, in planta it produces not only the toxigenic potential that the isolate has, but also it expresses this genotype in the phenotype. So we can see here, Meridionale produces NIF. The quantity is much smaller, but it still is important because NIF is known to be 10 times more toxigenic, more toxic to humans and animals than DON. Okay? Chapter 5, we are getting there. Man weapons. So we wanted to know which fungicide works better. So this is a good answer for the extension people. So you should know which fungicide is good, works better. So we collect data from many locations and years. We use a meta-analysis as a tool to quantify the effect size of these fungicides. And we could find that not only for, this is a box plot only, exploratory, showing that some carboxamides and tebuconazole 2 times was a little bit better than tebuconazole 1 time only. So the question was about spraying 1 or 2 times, also comparing these two the treatments of tebuconazole. And also the gain, the increase in yield, so the protection of yield. So we know that fungicides protective, they not increase yield, but they protect. So they work against the disease, okay? And then we could find that using tebuconazole, a second spray, we could get extra 100 kilos per hectare using a second spray after flowering. So the first one at peak flowering, and the second one 10 days after peak flowering. There was an increase in the yield, and the efficacy was not so high, was actually a little bit lower, so not different in terms of efficacy, but there was a little bit more protection when we use 2 sprays compared to just 1. And that's another one. We started to use, the first paper was about the DMIs, the triazoles, and the second paper was a mixture of QI and DMI. That's interesting because so many researchers have shown that using QI would increase mycotoxin. Some of the QI, some of the active ingredients would have a negative effect because there was an increase in DON. We did not test for DON in this experiment here, but we could find that a combination, a mixture of DMI and QI will work better than a single DMI. And also there was an increase in the yield here in some experiments. So not so different when we compared, but the idea is to use this to get this data, the yield increase here, so the return in yield, they are quite similar. So there's no major superiority of using when you use a mixture compared to a DMI. Okay, that's the take-home message here for yield. And show me the money. We want to see the money, so how profitable these applications are. Okay, when we have a more expensive treatment, a mixture is more expensive. Yeah, expensive than a single DMI. We have a greater probability of not offsetting the costs. So the blue is better, okay? The probability of not offsetting costs is higher in this yellow range here. So there was not so much difference because the best fungicide to increase yield is also more expensive. So it's just, so it pays off. If you use a not so good fungicide, you pay less, you get less yield, but it breaks even. That's break-even effect. And we get into chapter 6, failure now and ahead. We got isolates and we tested for fungicide sensitivity using P2 plates media for these two DMIs. In this case, we could find differences among these strains, but most of them were sensitive to DMIs. There was just one for tebuconazole. The EC50 was a little bit higher in one case from Paraná State here, and another one from carbendazine, which is a different group, one less sensitive here. So most of the failures that we see in the field in Brazil and many other places, not because of the lack of effectiveness of the fungicide. It's another thing. It's the protection of the heads, protection of the flowers. That's difficult, even for any floral disease. It's really difficult to protect those sites. So that means the fungicides work. They don't work in the field because of the operational aspects. And we tested when we compared the graminearum and also nivalenol, the meridionale ones, we could find that the dominant population, the dominant graminearum with the 15A phenotype was more insensitive because it's the dominant one. So it's more exposed. The dominant population is more exposed to the fungicide. So it's natural that the dominant population here for a DMI is less sensitive to the fungicides. Okay. So we learned a lot of lessons. Let me see how many you have. 8 lessons. One spray. The previous crop is not an important risk factor in the subtropics. I know that this is very, not many people would agree with me, but in Brazil, that's the case. So we cannot generalize, say no till is not important or grow wheat after corn is not important. Yes, because corn is a primary host for this pathogen. So we know that that's important, but in our case, it was serving fields that there's another word we did not show here. We could see stubbles, corn stubbles in the field and no headlight. In another place, there was no residue, lots of headlight. So it comes from elsewhere, not only from within the field. Because of the ability of this pathogen to travel long distances, because you know that they have aerosols, they're very, very little. At least 2 important species and chemotypes to be concerned about in Brazil. Graminearum with the 15-adol and meridionale with the nivalenol chemotype. DON and NIV should be a target in surveys, but only DON is regulated in Brazil. We have a regulation for DON, not for NIV. And one seems to be more adapted to wheat environments, graminearum. I did not show this data, but we surveyed corn in many places, and it's the opposite pattern. Meridionale loves corn. That's the reason I showed you. Wheat and corn more meridionale. In corn, meridionale is the main pathogen, and the mycotoxin is nivalenol. It's not DON. So if we are testing for corn in Brazil, we should be targeting meridionale. We should be targeting nivalenol and not DON. I think the situation in the U.S. is different.
That's what
you are lucky because you have the same pattern, the same chemotype across the 2 different crops. Breeders should use the most aggressive strains, which is good news for breeders. They don't need to worry about mixing or producing different kinds of knockout on different isolates. one spray of 10 conazole is a cost-effective choice in terms of yield. So it's not necessary to use lots of sprays. Just one targeting the full flowering, they do a good job. The premixes, the DMIQR, luckily do not break even costs because they are more expensive. They're not cheap fungicides. The pathogen may be adapting to fungicides. Maybe we should keep monitoring to see if with time, with more sprays of conazole, DMIs, and also QIs, there will be a concern in the future. So it's 5 p.m. I think I did it in 30 minutes.
We have time for questions.
Yeah, I just would like to acknowledge the key collaborators from many Brazilians, many colleagues from Brazil, and many students. And these people were very important during these first 20 years, because without them and without the students, I wouldn't be here presenting. So they did the hard work, I did the talk. Thank you so much. And this is the university, Federal University of Viçosa, the main campus. I don't know if you are aware of, but UFV and Purdue had an agreement in the past, 50 years back. I don't know if you remember that, but there was a lot of collaboration going on between Viçosa and Purdue. There's an avenue in Viçosa, in the university called Purdue Avenue. Purdue Avenue. And all the many faculty came here for training. Some faculty from Purdue came to work there. Yeah, it's very intense collaboration. And we have a project that we proposed, so hopefully this CAPS Purdue grant, we are going to make that work. Fingers are crossed. Thank you.
Yeah, thank you. I think that was very interesting to show exactly how you can build your career over those years. Like you start in a point and you go accumulating data and kind of. And I also, it's not because he's here, but I think that's something I always admired, how to create that and building those data. I guess a good organizing is important type of skills to have to keep track of all of them. But today, looking, of course, I know a lot of those words, but look at those data and the publications years, I can see the overlaps, like how it was built. So that is very impressive. We do have some people online, so thank you for the participation. If you have a question, please, you can unmute yourself or you can write on the chat. But also I'm going to open first for our public here, so in the meantime we can keep eyes on there. Okay. Yeah.
So I have one. You mentioned about coverage on the head and the flower. So in Brazil, are you having to deal with drone applications now?
I don't think so. They are not easy for wheat.
Okay. Well, I mean, we're completely shifting, and they don't understand what they're doing. So that's where we're going to run into. I mean, I'm looking here in the United States. The amount of drone applications going out and not acknowledging coverage is becoming a problem, and they're spraying fungicides all over.
Yeah, what's becoming very common in Brazil, as far as drones are concerned, is that people are starting to use a lot of drones, but there's no research.
We're doing
they
it
are
without
using
any
them for
research as
everything,
well. Right,
but
yeah.
there's no research backing up those results they claim.
Right, no, and that's what we're dealing with now, is they're saying they do better and there's
but there's no
data or no one's
research.
compared them.
No
Yeah, I just was wondering on that, because we haven't jumped into looking at the drones on our wheat trials. You just still do it with a ground rig and 45-degree angle sprays.
And it's not so difficult to set up an experiment and analyze the results based on the incidence and the index.
Right. Well, it's difficult for us to get the drones now, so that's the other side of it.
Okay.
That's a whole other gamut. University side, well, federally we can't use it. So.
You mentioned, I guess in the northern part of Brazil, they plant after late midsummer or after?
Late summer, yeah.
Okay. So they'd be following both corn and soybeans, just
Corn and soybean. Yeah.
whatever was in the field.
Yeah. Mostly soybean.
So, I
It's
guess,
the winter crop. It's the second crop.
yeah, but you're, everywhere you're growing, what in North America we call spring wheat,
It's spring wheat.
no fertilization.
No, it's spring wheat.
Yeah.
Yeah. Yeah. In the tropical area, there's a huge potential to expand wheat in that area. But the problem is wheat blast.
Yeah.
It's another disease. Yeah, it's interesting. I just gave a talk to Bayer yesterday, and about not to Bayer, to people at Kansas State about this wheat blast, and they have wheat blast group there, Barbara Valley. I showed this data that we have a short window for planting because we need to minimize the two hazards, the wheat blast and the drought. Well, the exception of the summer is the dry season. So if you plant late in the season, you're going to get lack of water. You're going to lack water. It's rain-fed wheat, you need water. But if you plant early, you have the water, but you have the disease. So we use a model to map the regions to optimize, to indicate the best wheat for planting to minimize both hazards. That's a tool that's a map that is good for extension that people are using. I hope the farmers are following that. Hopefully. We don't have an extension system like the University have here, so we depend, the farmers are on their own way of following recommendations from cooperatives or from companies, from the industry. So I provide these tools for free. We also have for wheat blast and for headlight also, forecasting system. It's an online mapping tool. You can easy access in your phone. You can just type where you are and you can see the risk for headlight and wheat blast. So yeah, Camille was mentioning about how to build a program. I'm formerly by training an epidemiologist, but with Fusarium headlight, I was lucky to have people and collaborators. So I have this opportunity to interact with many people in the US, to send my students here, to send the isolates, not the isolates, but the DNA. Isolates will be different. We can send isolates, but it's a nightmare. But if we have the partner, it's fine. I could send some isolates, also from wheat blast and headlight here. But yeah, to build a program, having a focus on the system, I was lucky because there was not many people in Brazil doing this kind of work. I was the first one doing the modeling first, and then it's like building a house. You have the bricks, and you just need to. And then I think I contributed a lot to this headlight studies in Brazil, and then I decided I need to move to wheat blast, but there was so many other questions there, and to use similar tools. So there's another movie if you want to listen some other time. I can talk about wheat blast. It's another movie with many chapters also, but it has been fun. So to do science is a lot of fun.
So we don't have to wait last year.
No, hopefully you'll have that.
Hopefully we don't have it.
Yeah, you don't have it. I don't know if you're going to have it because of the climate thing.
Yeah.
Not
for the tropics last year.
Not the tropics. Yeah, you need a knock-on first to build up during the winter, so I don't know if you're going to have that.
We do have one question there. What is the selection criterion for the Emerson test, given the isolates with the same complex exhibit different levels of aggressiveness? And then there is the other
What's the
question.
reason for large difference between design species here in Brazil? I think it's because it's a combination, I think, of having different species that are adapted to different crops. Like rice, for example, it's another story. It's not meridionale and it's not cardinale, it's Asiaticum from Asia. So rice came from Asia. So it's a combination of having the host and the weather favorable to for the epidemics to go on. So that's a combination. I think that explains a lot. So, but I don't have much clue about what bring those species. I can just guess that the host, that the movement of germplasm and the host seeds from different places. And we have a unique combination of species complex, different lineages of different species within the complex that coexist in Brazil and not in some other places. And first question is the selection criteria for the aggressive tests. Isolates within the same complex different levels. So we just pick some isolates that are representative of each species, and we can find some differences among isolates within the same species. So we tend to use a mix of species of isolates from the same species.
From the same species.
Yeah. And the aggressiveness test is just inoculation, and we have inoculated singles isolate or a mix of isolates from both.
We also quantify the number of spores, see how fast they can germinate across the species, and at least that's what I test.
Yeah, and also the fertility.
Yeah,
did not
for
show
the
me
purities.
Yeah, I need to make the look short. It's a lot.
Is there a known sexual stage for Fusarium or not?
It's the same.
Oh, it is?
It's the same, guy. It's the same. It's the
Gibberill Z.
Gibberill Z. Yeah, Gibberill Z is the same. We're gonna differentiate morphologically.
Yeah.
That's why. It's the same.
Yeah, it's the same. They just impact.
And it appears that based on the data for marriage for full time.
Yes.
So can you tell those two Fusarium apart morphologically, or do you have to use DNA?
DNA. Wow. Or you can inoculate the hosts of this guy looks like pattern. If you use one, it goes back from one, then now.
Yeah, those species with the Fusarium graminearum, they don't differ much. Like if you go across Fusarium verticillioides, then it has like microincognita, so you can kind of see the difference. But between the complex, no.
Yeah, there are so many species in the complex.
A long time ago, I think Vollenweber did a lot of Fusarium taxonomy. He had lots of species. And then Snyder and Hansen lumped them together just decades ago. So it sounds like it's going the other way now.
Yeah.
Yeah, they keep doing that.
The lumpers, right? The splitters and the lumpers.
Yeah, yeah.
Yeah, now they are splitting. The dominance is on the splitters. Yeah, they are splitting. But still, graminearum is the key species in the US.
Yes.
There are differences in terms of chemotypes. There are different populations like NX2.
Mm-hmm. Now in US, yeah, they are DAS and X2 is one of their another kind of
you had one slide talking about the efficacy of one of the, or at least multiple products on disease, and you said that one product in particular, maybe tibuconazole, had lower efficacy in disease management but higher yield rates. Now, did you take into account any other disease problems that were there that it might be managing, and hence, like, maybe there's less of a yield loss due to other problems that are occurring, or, why do you think that besides?
The treatments were applied after flowering, during flowering and after flowering. All the treatments applying to control foliar disease were the same across the plots. So it should be the same. There should not be more disease in one treatment than the other because it's sprayed against foliar disease. Different from the US, in Brazil they spray in some major high yield production areas, they spray up to 4 or 5 sprays in a week, which is a lot compared to the US. It's pretty much less. But because we have powder mildew early in the season, then we have 10 spot, we have rusts, we have hand lights. So it's a lot of sprays. And still they can make some money out of it. I don't know
how.
But yeah, it's very expensive. Grow wheat in Brazil, it's very challenging. Yeah. In the tropics, it's good because they irrigate, so they get to control the water, and they have high yields. So in the tropics, it's good. They are breeding for heat tolerance, also some different traits. but it's a place where we can expand wheat production in Brazil and to become self-sustainable.
Are these bread wheats or?
Bread, mostly bread.
You join here in this area, we were soccer meat, which
Yeah.
is donuts and all that kind of stuff.
Happy to have you here. I have a question regarding to the weather model. So were you using weather station, like on-site weather station, or it was like from a database?
To build the models, the original models, we use in station data. But to build the newer models that we just published a paper this last year, was using the NASA power data, because we were summarizing like 1 week of data, not a single day. So it was a summary, variable summarizing like average for the week, number of days of rain. We validated against station data, and it was doing the job. So it was not, there is error. Even if you use a station nearby, it's not going to be the same.
And you have to calibrate as well.
Yeah, it's expensive. You need to calibrate and maintain a station. So we decided to expand the not waste the data that we have, because using reanalysis like NASA Power Hero 5, you can get read data. You can use all the data points that they have. Otherwise, you would use just the data points close to the station data. So we would be limited in terms of data points.
And the other question that I have is about the ADPC. So I know that you just published a paper about proposing another method for ADPC. Can you talk about that concept?
Yeah, it's actually a different framework that you can fit any kind of function to the temporal data. So the function can be a logistic model, it can be a GAN, like a generalized additive model, it can be a spline, everything that provides a good fit to the data. And I'm calling that a phenotype. So that would be a phenotype. If you have a genotype, for example, a corn genotype that is grown across environments, okay, and you want to define what is the phenotype of that genotype, you can use AUDPC to classify that genotype in relation to other genotypes. You can use a Tukey test using AUDPC, okay, to group, to make comparisons among these genotypes. Or you can use the function. So you use the whole curve, so you don't summarize the AUDPC or the Ymax or the rate. So differently from using a scalar variable, you use the whole function. And then you can calculate the distance between two functions. Because it's easy to have the same AUDPC for two different curves. If the curve starts early and then stabilizes, it starts late and goes up, the AUDPC can be the same, but the curves are completely different. So you treat the distance between the curves. That's a mathematical formula to calculate that. So that's the idea of, I'm calling this functional disease phenotype, which can be used for breeders and also for crop protection if you have fungicides. And the fungicides provides control. There will be a curve. It can be a late surge, early control, early suppression, late suppression. You can use this approach also. But that's another movie, another story that I just published this letter to the editor. There are some other papers coming out in the next few years. I hope people find this useful.
So adding to that sounds exciting because I was just looking at the data. So say you spray a field, right? And you have 3 weeks of protection. And then the next, you sprayed a little later, 3 weeks of protection or double sprayed. Would it separate out those things and tell you the optimal timing depending on the disease development? You know what I mean? You know what I mean?
Yeah. Yeah, that would be very useful for this kind of a situation. You get the protection early and then
yeah, or where does it run out, or where did we miss it, right? So if we
Yeah.
went too late, we missed it. I guess I'm just looking at the data set that I'm trying to get my head around. And
Yeah.
now you just added another layer of work.
And you are not limited to one-time assessment. You have an entire curve and you don't throw away information. You just by using one style, for example, like the UTPC, you use the whole curve. Yeah.
You see the whole scenario.
Huh?
You see the whole scenario.
Yeah.
Yeah.
For questions, we don't have online. Another thing I want to mention, maybe we can share later, but another important component that I always admire that he has everything online. So, and it's open access. So you have multiples of.
I think I gave a talk here.
Yeah.
Yeah.
Remember
Oh,
2 years
yeah.
ago
Yeah.
about this open access? Yeah, I
Yeah.
gave a talk to this
Yeah.
one.
It was like an online
Dr. Ponte
seminar.
back in- yeah.
Oh, okay.
Yeah.
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 They are aware of that.
And I have my plant disease epidemiology course going on this in August. It's online, it's English, and it's free for everyone. So we have applicants from many places, most from Africa, more than 50 people. It's only epidemiology, plant disease epidemiology course, taught online and free in the world. I think it's the only one. Yeah. Because I have the book, Plant Disease Epidemiology, the course is based on that, which is the online free book, which is evolving every year. It's not a static book.
Yeah, they have a lot of like really useful code for code AUTPC, meta-analysis, disease evaluations as well. Disease is measurements or scales as well. Yeah.
Well, if nobody else has questions, I was just gonna thank you, and
Thank
that's
you.
a little Purdue